Welcome everyone to our office hour today based on clustering and taxonomy building. We're from the Allen Institute for Brain Science. I'm Rachel Hostetler. I'll be moderating this office hour. And today we are joined by Nelson Johansen and Dan Yuan. And I'll allow Nelson and Dan to introduce themselves.
Go ahead, Dan.
Yeah, hello everyone. My name is Dan Yuan. I'm a scientist in the brain science in the genomics team, specifically in the data and the informatics team. So I've been at the institute for almost 2 years. My role involves helping build a whole human brain taxonomy with a focus on understanding the conservation between human and mouse, so that we can leverage the well-annotated ABC atlas to annotate human cell types. On the other side, I have also been working a little bit on the methods development side. For the goal to be able to analyze tens of millions of cells from the single cell data, we need to have the methods that is scalable enough to be able to analyze those number of cells. So I have been kind of improving these two methods that the scientists at Allen Institute have been using, or reinventing certain methods to deal with problems like donor effect doublet detection, as well as integration across species like human and mice.
I'm Nelson. I'm a scientist here at the Institute. I've been here for I think almost 4 years now. I'm on BICAN's team, and we're really a cross-species first kind of team, looking at evolution and conservation, and building taxonomies with a multi-species view from the get-go. Along with taxonomy building, I do a lot of deep learning modeling and kind of standards for helping other folks build taxonomies.
All right, thank you, Nelson and Dan. We can just start with some general questions. I have a couple here, and then if anyone wants to jump in, ask any questions live, you can enter them in the chat. But first, we can just start with broadly, what exactly is a taxonomy? What types of taxonomies are you working on? Why are you building these? And, what kinds of information can we get, from these taxonomies?
Well, I can take the first part of that question. What is a taxonomy? At least in my mind, and Dan, feel free to disagree with me. But at the Institute, we have a cluster-centric view of taxonomies where we, you sequence a bunch of data from either brain or really any tissue or organism, and you get cells, right? And their gene expression, but then how do you organize them into like things? And at the Institute, we use clustering to group cells together. And clusters are a minimal unit. So as we build a taxonomy, it's taking all these, thousands of clusters, and how do you merge them into, classical cell types and eventually a hierarchy, which I think is really the core part of a taxonomy, is this, course to really fine annotation of what cells are doing molecularly, and hopefully embedded information from other modalities or other, literature sources. Yeah. You had a few questions in there, Rachel. That was the first point. What was
yes. Sorry, it's all kind of grouped together. So, I mean, yeah, you could briefly mention some of the which species or which taxonomies you're working on now, and what type of information do we get from these taxonomies? Why are we building them?
I can jump into that one. I think so with this human mammalian brain atlas project, the Allen Institute has been working on for a while now. We're looking to build whole brain atlases of human, macaque, and marmoset, influenced by the mouse whole brain. Dan mentioned that she's working on the human whole brain part. But my efforts have been focused in basal ganglia for a while. Taxonomy building is difficult. And it's even more difficult as you bring in multi-species and inter-individual effects. So, we're all unique, haplotypes, mitochondrial and all that good stuff from, it makes us, individuals. And that leads to batch effects in data. So we've been figuring out how to handle these batch effects while building taxonomies for whole brain, which the mouse thankfully doesn't have to deal with, given that they're isogenic. But we're learning in basal ganglia how to do these primate whole brain atlases and building out the tools and the foundations. I think one really critical thing that we're doing now with HMBA, and folks have really advocated for this, is trying to fold in literature at every point they possibly can. We build taxonomies that are sometimes disconnected from previous papers based on the names that we use. And I'm really trying to make sure that we build taxonomies the field can use by embedding previously well-used names for cell types. In basal ganglia, an example is these islands of Calleja. They've been explored for decades, and who are we to rename that cell type? So trying to embed those kind of names at the forefront, I think, is really critical.
Yeah, Dan, and feel free to add on if you have any thoughts on that. And the data set's primarily transcriptomic data, single cell or single nucleus sequencing, correct? For most of the work you're doing. I know some of the work uses multi-ohm, so looking at ATAC-seq, but the ATAC-seq data, how is that used in regards to building the taxonomy, or is it used at all?
You want to take that one, Dan?
Okay. Yeah, I can add later. Do you want to go first?
No, go for it.
Oh, for the whole human brain taxonomy I've been working on, we just use a transcriptome to define the cell types first. But this is a multi-ohm data we collected, and I imagine we will use the ATAC data combining jointly with the ATAC with the RNA-seq to have a joint reduced dimension space and then define cell types, and use the ATAC-seq to find cell type specific cis regulatory elements, and then or enhancers, and then the transcription factor regulatory network. That's just will be the part of the analysis downstream.
Definitely agreed with what Dan said. But keeping in mind that every molecular profiling kind of gives you a different view of the cell. So when you cluster RNA-seq, you get a expression-based kind of landscape of what a cell type is. ATAC-seq is difficult to work with. You're working with whole genome level features, which is super high dimensional. And it turns out that cell types that are very distinct based on gene expression are not very distinct based just on open chromatin. But that said, you get different axes of variation from the open chromatin you don't get from the RNA-seq. So it's how do you handle those two scenarios at once? It's a difficult problem. But I view ATAC as really useful because you can kind of sanity check your cell types for the most part. An RNA-seq type should have open chromatin distinct patterns. It's also a way to compare across species. If the RNA and the ATAC profiles agree across species, it's probably the same cell type, and you can find that link. That's one area I see ATAC as being, I think, really useful. Because genes, they aren't as conserved as we would like across species, but it turns out the genome and its openness patterns are a bit more easy to work with.
And then, especially with the whole mouse brain, now we have spatial transcriptomic data that's complementary. And I know there's some of that in progress with HMBA as well. And then there's also things like, you mentioned cell type naming, right? So there are people who have said these are fast spiking cells, or these are low threshold spiking cells for decades. So it's like, how do you, take other modalities, like especially electrophysiology or, spatial transcriptomic data? How do you take that into account, or is that being taken into account when working on building these taxonomies?
I can speak from the HMBA side of things, the basal ganglia work that's, it's all going to be published here in August. We've been very closely working with our spatial folks. So with basal ganglia, we have MERSCOPE and Xenium or Xenium for human, macaque, and marmoset. And we take our taxonomies, right? We map that into the spatial transcriptomics to get where our cell types are spatially. And it turns out that's a really useful piece to have because it's hard for us to know where these cell types are sometimes based just on the dissections. So from the molecular side, we just know like where the tissue was cut out of. And while we have experts doing this, it can get a little fuzzy sometimes, but you kind of can't, the spatial transcriptomics helps us really nail in where these cell types are localized. So we've found with HMBA that it's really an iterative process. Once you have clusters, you map them to spatial and you help inform each other on how to describe these types. The same with patch-seq, how do you find the fast spiking cells? Only the ephys can tell you that. It's easy to do with these molecular measurements where you have RNA-seq to latch onto. Previous papers that only did morphology or physiology, that's a bit harder to find that link. But I would say it's invaluable to have at least spatial transcriptomics when cell typing and have a really tight feedback loop between modalities. I don't know if Dan, you have any?
Yeah, totally agree. I just want to add one point that from the ABC Atlas, the Nature paper in 2023 by Yuan et al., the spatial data is extremely important to help validate the clusterings from the transcriptome data. It really helps flag some of the jump clusters and pinpoint where those cell types are. And if a cell type is distinct, it's likely that they will have a distinct spatial location.
I guess to expand on that a little bit, with the RNA-seq, there's clear islands of cell types, right? But then you have these continuums of cell types that look unclear based on just gene expression. When you map them into the spatial context, it's immediately obvious that these are one type or the other. So you kind of can't solve the whole taxonomy problem with just RNA sequencing data, in my opinion.
Right. That I was just checking. You weren't on the were you involved with the whole human brain study data at all, Nelson?
I wasn't involved with helping make the taxonomy. We use it all the time.
Got it. I was going to see if you had any thoughts about the splatter neurons.
I love the name. I think it's a very apt name because, even with HMBA, with this whole brain across species, we still see a group of cells that looks like a paint splatter on a wall. And it's an apt name, but it's also not a very useful name, right? I'm not sure you can go back a decade and search splatter and get anything. So we went the mouse whole brain work. I know, Dan, you've done some of this, and maybe you can talk about it, but you've been kind of parcellating out the splatter cluster into some real cell types.
Yeah, we have been utilizing the ABC Atlas because it's well annotated for, of course, for the whole brain, but also a better annotation in the brainstem area, the splatter region. So kind of kind of use the ABC Atlas to help us annotate the splatter region that was in the selected taxonomy. And we do have a better resolution in that region now.
Yeah, I think there's some really amazingly well-studied cell types in the splatter type. A few examples that I at least know from the basal ganglia work are GPI core and shell. These are types that literally form a core and a shell in the globus pallidus internus. And those are called splatter, along with these islands of K I was talking about a little while ago. Those are in the splatter group, and it's a shame a little bit that these types aren't kind of brought forward, but it's also a first version of the whole brain work, right? So we're building off of them.
Yeah, and to provide some more context for these neurons, so in 2023, there was a paper package published in Science. In the paper that we're talking about, Saletti is the first author, and it was the first whole human brain transcriptomic atlas. And with the clustering, after all the clustering, if you look at the T-SNE plot, there is one area of the plot that legitimately looks like a splatter, and there's a number of different cell types that are within that splatter, but the overall large group, the supercluster, is called the splatter neurons. But then if you go look at the anatomical regions, see where they're from, there's a lot, from the pons, hypothalamus, amygdala, some other areas that Nelson mentioned, but yeah, they're just called splatter because that's just how they looked when you ended up doing the clustering and plotting, which is kind of funny.
I think our goal is to, like Dan was saying, like figure out what's in the splatter and also what's in miscellaneous. I think that's another term from Slutty et al, which understandable given that these types are all very kind of on a continuum of expression, but we're trying to take these terms and kind of parse out the better nomenclature for them in the HMBA and BICAN work.
Yeah, and you can view the taxonomy and look at the splatter. If you go to ABC Atlas or Allen Brain Cell Atlas, and if you go to the whole human brain taxonomy, that's where you'll find that. Or it's called transcriptomic diversity of cell types in the human brain. That's its data file title. But then yeah, you can see the splatter neurons yourself. And this particular project it's really the first version, right, of the whole human brain atlas. And there's a lot of work underway at the Allen Institute and with collaborators to sort of do the next version of this atlas. So yeah, it's a funny name though.
It definitely is. And we're also overusing the term whole brain with Saletti, right? It's
A little
definitely
bit, yeah.
definitely a great coverage, but not whole brain. So this next effort that's building off Saletti et al is much closer, I think, to whole brain coverage, which is really exciting, I think.
That's true. There are a few areas that were definitely undersampled, which are, if you end up reading through the paper, you can get more details on that. But first version. Yeah, and so I guess I'll go back to we went off track a little bit, but that was my fault. We can go back track to talking more about just taxonomies in general. So when you look at like different taxonomies, especially like the human brain Saletti taxonomy we just discussed, and then like the whole mouse brain taxonomy, they have different levels of division and different names, right? Like the Saletti one goes supercluster, cluster, subcluster, and whole mouse brain is class, subclass, supertype cluster. And then I've seen maybe in some of the newer stuff, there's another level that's added in called groups. So what are all these different levels? How do you define them? And then, how do they connect perhaps, when you're doing the cross species work, right? Like different names or different types. And how are these determined? Like marker genes, how do you figure out what goes at each level?
Dan, do you want to go first? I know from the mouse side, y'all have done some really cool work with like TF clustering to find certain levels that I haven't had a chance to poke around, but I'm really interested by that idea.
Yeah, I can maybe talk about from the mouse side, what are the four levels, and can start from there. Yeah, so from the class is the most coarse level where there are like 30 of them in mouse, and we found 30 of them in human as well. And it's often named after the broad brain region plus the neurotransmitter type. And then a level below that is the subclass level, and then supertype and a cluster. So the levels are built after we do the de novo clustering first. After we get the cluster IDs, we use a transcription factor to group the clusters into class and subclass under different resolution. And then, and then the supertype is built within each subclass, and then we group the clusters together to get a supertype. And for either subclass or supertype, there should be a very distinct spatial location for them. There's a very distinct pattern for them in the spatial coordinates. But for clusters, some might have, some might not have. But subclass is the one that you should find a very distinct spatial location from the spatial data. And if you go through the 2023 Nature paper, you will see that the authors have gone through very thorough and comprehensive literature review to establish those 4 levels for the mouse atlas.
And then on, I guess, another viewpoint on this level thing, I think to be honest, we've had a kind of convention of classes broad and subclasses well-defined, and then clusters are your finest units. I think historically we defined subclass based on types that have very well-defined marker genes, which then implies that someone who wants to map their data to a taxonomy can kind of do it reliably at the subclass level. You mentioned the group term, Rachel. This is a new term we're bringing into the HMBA basal ganglia taxonomy. And, it's one of the hardest things that we do is trying to find homologies across cell types, across species. And this new group level that sits below subclass, above supertype, is a space where we are essentially completely confident in the fact that this cell type is conserved across human, macaque, marmoset, and mouse. And we can use the exact same name for all 4 species. And then the same as you go above group. So subclass, class, and neighborhood, those are also all conserved. So we're trying to put kind of a line in the sand on where we're confident in describing cell types across species based on a level in the taxonomy. But it's a lot to learn, and it's a lot of levels sometimes.
Yeah, so with that, so if you compare the super types across, macaque and human, for those, you're not going to see the one-to-one relationship. But at the group level and above, well, maybe not one-to-one, but you'll see whether if it exists, in all kinds, it'll be in all the different species. So are there certain cell types that are more difficult to compare across species, especially when you get up into human and non-human primate, right? You have different ratios of neurons and glia compared to, looking at the mouse. So when you're doing this large cross-species work, are there certain super types or certain types of cells that are just hard to compare across species?
I mean, maybe the cop-out answer is just the rare cell types, because you can't collect enough of them really in any species. An example of that is the dopaminergic cells of the substantia nigra. We have a hard time collecting enough of these in primates to really do good cell typing without enrichment. You have 2 kinds of sequencing, unbiased, where you maybe sort loosely for neuron and non-neuron when you sequence the cells. And then you have an enrichment strategy where you have a known marker gene and you try and only take cells with that gene expressed. I think the cases where we have a difficult time is those rare populations. When we do these computational alignments, it's when you have rare cells, you're not sure if the alignment made a mistake or if it really is a good agreement across species. That's where I view some of the difficulties, at least.
Yeah, I also found there is a population in the brainstem. It's so hard to integrate between human and mice, and no matter how I think I kind of push the limits of the current methods to trying to integrate them, but they still refuse to integrate. And if you do label transfer, they won't be transferred to the same label. So that could be sampling issue that we're just not sampling enough cells from that region for human. But we're also pushing to trying to improve the current methods to see if this is something we can do from the computational side. But yeah, certainly, I think the integration is a lot harder in the brainstem region because the cell clusters are so tiny and they're so distinct.
Yeah, oh, you can go ahead, Nelson.
A little step back. We keep saying integration and alignment, but I want to make sure that we clarify this. We probably should have done this earlier, but we mean you take multiple data sets and try to find a common representation of all the cells across those data sets in an embedding space. Different species, you have variation along the expression axis that kind of separates cells. So methods like SCVI, Seurat, it's a whole slew of them, allow you to bring data together and put them into a common space where the same cells of a similar expression pattern or cell type grouped together. And allows you to compare like human and mouse and other species and other studies. Just wanted to clarify that for folks.
Yeah, and I wanted to mention that this top brainstem came up a lot in our previous office hour, just when doing the actual sequencing and preparing the data. I learned that especially with human and non-human primate, it's quite difficult to get cells from that area just because of the amount of myelin and debris and other things that interfere with the actual process of isolating those nuclei. So we heard, yeah, in our previous office hour, if you watch that, we can get into more details about like why, at the technical level, when you're collecting the tissue, collecting the nuclei, why you lose or have so many issues, getting cells from the nuclei. It seems to be quite a challenge. And then you can see that, once you get to the data, that you see that reflected there.
Yeah, and certain cell types don't take to the whole dissociation sequencing process well in the first place. I'm sure medulla and definitely in spinal cord where you have these massive motor neurons, they don't like being spun and dissociated. You can really destroy the cells pretty quickly. So there's biases when building taxonomies that start at the very beginning.
Yeah, that was also something we discussed a bit, right? Like when to use single cell versus single nuclei, sequencing and, the whole mouse brain. Most of it's single cell, but there are a few areas, I think also in brainstem or midbrain that are, came from, they had to collect the nuclei instead, right? Because the cells were not compatible for that process. And then all the, the human and non-human primate work is nuclei. So yeah, a lot of details. But from what we learned from the last office hour is that there's also been a significant improvements in doing the working with nuclei and getting, good sequencing data from that. And that there's been a lot of improvements in the last couple of years. And that, since we use both types of data at the Institute, the scientists and those working with the data know how to compare the two and know how to, integrate them and work with them together. So I guess, yeah, any other comments on that? Working, sometimes you have to work with both single cell and single nuclei data.
It's a pain. I mean, you see we can do it, but it's still difficult, right? With single cell data, you have all the cytoplasmic reads, which you don't get with the nucleus only. So it becomes a really difficult problem trying to align them together. I know the mouse whole brain work is going that direction. Dan, you can probably talk more about that. But I think it's easiest when you have the same modality, for sure. And our colleagues, Rebecca Hodge and others, have really optimized single nucleus for primate tissues. Single cell just doesn't work, as far as I'm aware.
Okay. So some more questions that we have. So we talked a little bit, or this came across earlier, looking at rare cell types. How do you really determine when something's, novel or rare versus junk? What's the process that you go through to help determine that difference?
Oh, I haven't gone through that route yet. I was like hoping to use the spatial data to help with that. So also maybe a better position to just that.
Yeah, I think leveraging a few things when you have spatial, that's amazing. When you don't have spatial and all you have is RNA sequencing, I think one of the most useful tools we have is that we sequence from multiple donors and multiple libraries. Um, a rare small cluster only from one donor and one library might be real, but it also might be junk. But when you have multiple donors, multiple libraries, all kind of telling you that this cell type was sequenced from them, that's a really strong indicator that that's a real entity and not junk. So on the primate side, we usually use metrics based around for every cluster, is there enough donor support or enough library support? With the caveat that we know a few libraries are the only libraries we have from small nuclei. So trying to use the things you have, the metadata you have at hand to help distinguish those two things is really kind of the best thing you can do. I think one of the strongest assertions that we can make with multi-species is if you see that cell type or that cluster from multi-species, uh, that's almost an assured like that is a cell type. There's something interesting going on there. You wouldn't expect to see maybe the same junk pattern across species. It's a hard problem though. I mean, easy to be fooled.
Yeah, I know that also with the Alzheimer's taxonomy, which I know neither of you are part of that project, but that's also a question people had, right? How do you know that this was truly a novel, microglia that was only present, versus something, versus junk. But yeah, I think when you have, a large enough sample and you can see it's not just from one donor, that's definitely an indicator that it's likely, something meaningful.
You've seen fun cases in the past. The inter-individual study that was out in Science a few years ago, we actually had cancer cells from one of our human donors. And that is a unique island in the latent space. And we thought it was a cell type, but you look at some of these, cancer markers and it's obvious what it is. So it's not just junk, also can be other properties of the donor that you sampled from that could drive unique clusters or cell states in that case. So fun little example.
Yeah, that didn't cross my mind that that would happen. Again, I come from a mouse background, so in my mind, they're all the same genotype, they're all this, they're all that.
It sure makes
Yeah.
life easy.
But yeah, a lot of much more variability when you're working with human tissue. Um, yeah, so with that, are there, what type of, quality control measures are you taking? I guess this is sort of related to, that question of determining what's a real cluster versus something else. What are some, quality control measures that you take? And, is that like also species dependent? Are there extra steps you have to take when working with, I don't know, perhaps macaque or human versus other species?
I think we have a general sense of the basic quality control, and I know Dan, the mouse team, has done some really amazing work here on defining like scores for QC that include, how many counts, how many genes, doublet estimates. There's doublet detection algorithms. We measure a lot of data by the Institute. How well the reads align to the genome is another indicator of quality. And we try to find the best way to use, at least at the start, like total counts, total genes, doublet score. How you use those, I think, is really important. You could just do like per cell, flag them if they like hit your threshold, like double score greater than 0.3. That works. But the problem in my mind is that there are cells that are just above that threshold that kind of squeak by as oh, you're good, but maybe not. So we do both single cell and cluster based QC, where you take a cluster, you kind of assign it the median of the doublet score, the total counts, total genes. And then you can kind of do a guilt by association. If a cluster is like 90% junk based on your thresholds, you throw everything out. Um, I found it to be a really useful technique because it just cleans things up a little bit faster. Um, yeah, Dan, do you have any thoughts?
Yeah, I think we have been using the same QC criteria in the pre-processing steps. And then after doing the clustering, we also do QC at the cluster level. And that's where some of the manual inspection comes out the way. Yeah.
One thing, that we struggle with quality control, is that not every cell type or whatever entity you have in your system doesn't follow the same kind of QC criteria. We've defined some like really good cortical QC criteria that just work. But as you move whole brain, like, the cerebellum, you have granular cells that are itsy bitsy, and they just have so few genes, so few, UMIs or counts, they kind of look like junk based on your cortical thresholds. So it's really important to make sure that you have some way to know when you're throwing away real cells with unique properties. On the flip side, motor neurons and spinal cord, they're massive, and they kind of hit that upper limit of counts and genes for our QC threshold sometimes, and you could throw them away by accident. So I think we're still learning how to do this whole brain, but I think this applies no matter what system you're working on. Like, not every cell will have the same molecular or physical properties, and you have to handle that on QC side, which is really difficult.
Yeah, we can definitely see that depending on, which area of the brain you're working on, especially human brain, there's huge differences. And, even just acquiring, your nuclei, going through the steps, getting data from it, and then the type of data that you get. So it sounds like there's a lot of troubleshooting, at different steps. But because we started in, a lot of the original work was done in cortex, right? So that was sort of the baseline for that. And then you start going subcortical and see all these differences.
One maybe shameless
Oh.
plug is that Dan and I have been, and others, have been working on a package for QC called SciDunk. It's a public GitHub repo. Um, it's meant to help us, at least internally, track as we do QC in a way that we can always roll back and see like what was thrown away. At least I, mapping all the cells to reference taxonomy. Then when you throw cells away based on QC, you can go ask the question like, was any of the reference cell types thrown away in a biased manner? So we've been trying to be better record keepers of our quality control process. And I think everyone can really benefit from that.
I know that, when you read the numbers from the whole mouse brain, there were close to 8 million cells originally, and then about 4 million made it through, the quality control. And so that really, I mean, that speaks to the amount of, inspection and control, and just looking at, each one that only, the ones that made it were very high quality. Um, okay, so sort of going back to taxonomy building, um, could both of you sort of, lay out the different steps of really creating a taxonomy? So, starting from the data that you first get, going through the process, building the different levels, and, you know, are there different types of clustering methods that are used depending on which project that you're working on? I know we have some of the, a lot of the packages, or a lot of the clustering that's done is used with some methods that were made in-house. They're on our GitHub page, but yeah, general explanation of once you get the data, how you build a taxonomy, and if there's any differences in methods depending on which project or brain area you're working on.
I can start, I guess. That is a fun question. I think it's a complicated question to answer because we try and balance kind of at the beginning, doing a rough clustering that helps us pick out low quality and remove those before we do these really expensive high resolution clusterings. But I think we kind of have a workflow for taxonomy building set up now that, folks in our team who aren't computationally like first are able to take and really build a great start to a taxonomy. I have not summer intern, but a visiting resident, a visiting scientist who has never built a taxonomy before, and kind of 3 weeks in, we're kind of at a clean, whole macaque brain taxonomy. I think it's mostly due to the fact that we have kind of put together a standard. I think, at least, I don't know if Dan would agree, of how you kind of march through the easy steps, which is, quality control, clustering, and just handling batch effects and all that good stuff. That's all the easy stuff. Quality control is less easy, but still manageable. I think a lot of the time is spent really in how do you assign a cluster to an appropriate name, which is not a well-defined process at the moment. We hope that like large language models can help us. If you have a cluster and it's mapped to like 10 references, how do you make sense of all that? We're hoping to use like AI and LLMs to help sleuth through all that data and make a reasonable like guess what that cluster is. But for at least getting to a spot where you can annotate, I think we defined a good path to get there. I don't know if you agree, Dan.
Yeah, totally. I think we sort of have a shared standard way to build a taxonomy. I haven't involved in that much in the like later cleanup and renaming all that stuff. I've been through a little bit, but maybe not as that intensive as you have done. But I think we sort of a way to build a taxonomy from the raw counts from the very beginning to the end. And yeah, we have been consistent in all the clustering parameters and the QC parameters. And I think for me, the process I've been using is like the codes are public on GitHub and everyone can use it. So yeah.
Yeah, at some point we can have a like template notebook that we can have on GitHub that's polished and the folks can use it in the community. We're kind of talking like taxonomy building in a single species. I think it's much more complicated when you have multi-species. For the HMBA basal ganglia project, we ended up going through the standard steps and clustering each species independently. And that is a nightmare to deal with because now you need to sync up, 3, 4 species clusters with each other to start building annotations. So, going forward, thinking of kind of flipping the script where we cluster across species first and get that like conserved level and then dive into the high res clustering from there. So, just to say that we're not set in our ways either. We're still finding ways to build taxonomies as data gets larger, more pieces are in play. It's definitely an evolving process.
And then you had mentioned, always referencing, previous literature, previous cell type names. Has there ever been a case where you have something and based on your transcriptomic data, it's not in agreement, with the previous cell type name, right? Because a lot of cell types were named, using a lot of antibody labeling, right? A few decades ago. And obviously when you're looking at RNA, you might find something different. Has there been like ever a time where you're like, oh, it was called this, but actually it's sort of like split, it's sort of it's really two groups, or it's actually like sort of these two groups combined, or just something that's, not in agreement.
We see it a lot. Even the mouse whole brain versus, the slutty doll, right? Like what we call a subclass in mouse whole brain doesn't align perfectly to any one level in Saletti. So there's definitely splitting going on in taxonomies. And then, yeah, out to literature, you have folks who talked about medium spine neurons for decades, but there are many medium spine neuron subtypes that exist transcriptomically that we can identify now. So reconciling that's very difficult. Yeah.
So it sounds like it's mainly, maybe groups that were considered one, but really there's multiple groups within that group, as opposed to something getting an entirely new identity, or just, yeah, or like you haven't seen anything where something like completely like just doesn't make any sense at all. That's but it seems like it's more just like, oh, it's just actually this group has really three groups, or this group is actually, a bit more diverse than was thought.
I think a good example of this is maybe not from our data, but from Evan McCosco's lab. They're really interested in the dopaminergic neurons. They've done enrichment studies of those cells. So they've enriched for dopaminergic neurons and sequenced those. And they can show that like, R1 dopaminergic group really is like 10 when you have, tens of thousands of these cells through an enrichment, which we haven't done. That's an example of like one group becoming many from someone else's work. But then like, back to literature, we have a hard enough time linking our work to like the really old studies that don't have any like gene expression measurements to kind of latch onto to find that link between our work and a paper. So I'm sure there's many cases where we're talking different resolutions. And then naming's a whole nother thing. I think we're trying to put forth with HMBA a consistent naming standard that does the best job in folding in literature terms, but we're also trying to kind of put the final name out there with a kind of a really controlled scheme for describing a cell type. And that by nature, like adds one more name to the field, but hopefully is a consolidating name. If that makes sense.
Yeah, and I think being aware of aliases, or having some place to keep track of them is also crucial, right? Because there are people who've been studying this one cell type for, 30, 40 years, and all of a sudden, it has a new name now and new properties, and
I
yeah.
It's such a complicated problem. Like for basal ganglia, right? There's literally, I think, 10 or more different ways to say direct pathway medium spine neuron. And it's a pretty coarse level. So we definitely need to keep track of how folks have talked about these types. And I don't think any one person or even a collection of people can do this. I think this is a problem for, LLMs that can find associations and talk to someone. We all have our own way of describing a cell type. And if you can talk to an LLM to tell you, hey, like I call it D1 MSN, what's it called in the taxonomy? That's the only way to like ease people into new nomenclature, because it's such a complicated space.
Yeah, I'm excited about that part, and potentially using, yeah, AI and LLMs to help connect that. Because yeah, it's a lot, especially if you're looking at some of the papers from many decades ago, and they're really important papers, and then you're trying to connect the dots between, well, what is that, cell type, today, or how does that connect today? Or even just between some of the Allen Institute taxonomies, right? Like they're especially looking at mouse. If you compare the 2016 visual cortex to the 2018 visual cortex and part anterior lateral motor cortex, and then 2021 whole cortex and hippocampus, and then whole brain. Even between that, right? Like because I was in graduate school during this time, and I was trying to piece together, cell types I studied between the different taxonomies and seeing, where they changed, where they were consistent, because they're given different names. And so I think, yeah, even keeping track of cell types through our own taxonomies can be a bit tricky sometimes, and is something to pay attention to.
Yeah, definitely a difficult process. I think literature is really important, but I think it's important to be mindful when you build a taxonomy of what's been described for a given system when you kind of start down this road. For basic Anglia, we did this, Yuan, others and I tried to read the field, which is an enormous task. And I don't see it happening for an individual group to do that kind of going forward to whole brain. So we're going to start actually running these community jamborees where we actually invite external experts in who can, inform, embed knowledge, and help like distribute that load of knowing the literature, knowing the history, and embedding that into a taxonomy. So I think we're kind of trying to push into a new space where we really get community feedback much earlier along in the process. And define synonyms, define aliases, define literature links.
Yeah, I think that's super important for a number of reasons. I mean, one, because it's hard to say this is the new taxonomy, use it, and then people are like, well, I don't necessarily understand, what this means, what these terms mean. But then also, yeah, to get, feedback from other people to get, to build it to make it more accessible to everyone, right? If they're used to certain cell terms. And I think community involvement like that is so important, especially for these very large open science data sets, right? Because they're being built for the community to use. And we want, people to be able to use them and understand them, and for them to make sense. So I really like the idea of having those community jamborees. We have a couple more minutes left, so if anyone has any, questions that they'd like to ask, they can ask them in the chat box. If not, we will take a moment, and we'll have another office hour in June. We have not announced the date or topic of that office hour yet, but we'll post that up on our website. But these office hours have been a new format for the Allen Institute, just to try to engage with some of our external scientists and some of the external users of our data. And this will also go up online on our YouTube channel afterwards, so that others can watch this recording. All right, so it looks like we don't have any more questions. Thank you, Nelson and Dan, for being here today. Thank you for answering all these questions, and thank you everyone else for joining us.
Thanks for organizing, Rachel. This was fun.
Thank you, Rachel.
All right, see you.
Yeah.